Okay, I accidentally clicked stop recording or something with my pen when I was doing it too high up, I think. So this is the uh, DNA. I'm going to do replication and transcription, translation, transcription, not in that order. Okay, so I'm going to start over. I have my DNA template, and I should have probably just drawn this out before I started my um, drawing again, make sure it doesn't. Before I started recording, I don't know where I started with. I had different letters before, and that's what's cool about this. You can grab any sheet of paper, and you can make out a strand of DNA as long as you're using these four letters GTAC. Okay, and you can just make it as long as you want. I do recommend you count and try to make sure that you're doing enough that you can divide these evenly into sections of 3 later. Okay, I think that's, yeah, okay. All right, I'll change color for you. And then I'm going to come down here and we're going to do the cDNA, the complementary DNA strand. G bonds to C, T bonds to A. Okay, so that's what a complementary strand looks like. This is all DNA still. So that's replication. Okay, so now we want to make a RNA strand. And on a short answer version of the test, if I were to give you a short answer version of the test, then I usually ask that's the next question. Next question is, okay, make an RNA strand. And the problem with making an RNA strand is that if this cDNA strand is here on the paper, you've written it out, then it's really easy to actually accidentally make your RNA from this strand, and you cannot do that. You have to go back to the template strand. So I'm going to undo this, I think. I think it might be easier than erasing it. Oops, if I don't accidentally shut the, I don't want to clear all drawings. Okay, so yeah, I'll just keep doing this. Accidentally shut the program off again. Okay, so I'm going to take my pen of a different color now and come here and we're going to make the messenger RNA. Okay, so G bonds to C, C bonds to G, same way, and T bonds to A because A exists in RNA, but A doesn't bond to T this way, going this way. T gets replaced by uracil. And I didn't see that written out actually on any of the notes. So I'm going to go over here and write that up here in the corner for you. Uracil. We had the U there on a note, but what it stands for, I don't think was ever written out word-wise. Okay, so, and then we have a G here and a G here. And actually what I recommend at this point in time is just pause this on your own sheet of paper, finish this strand out, and come back and check it. Okay. And I'm going to continue on here. Of course, as slow as I'm writing, you might not have to pause it. You can just, go zip on down to the end and then wait and watch me catch up. Okay. Did I make a mistake? I did. Look at this right here. So yeah, always go back and check yourself. Should be, what should that be? You know, you. Okay. Be that proofreading enzyme. All right. That looks good. Okay, so now we are going to read our codons and make an amino acid chain. Okay, so I'm going to bracket that with black. You can go through like this. I have students who do this. Okay, and mark off all three. I don't care. That works really well. Okay. I do it a different way because I was taught a different way. I like it a different way, but I don't care. Okay. And I'm not going to see your work on this anyway. So you can designate your three codons however you want on your scrap paper that you'll be using. Okay. There we go. All right. So there's my brackets of my three, not neat brackets. Actually, is as difficult it is for me to write on this, it might have been better for me to do the slashes, but that's just not what I'm used to seeing. So it's not what I'm doing. Okay, so now here we have to use a table. So I'm going to try to get out of this without shutting off the program. Okay. I'm paranoid about doing that now. And let's back up. And of course, my writing is on the table. I don't want to do away with that. Yuck. All right. So our first, if you can see it kind of through everything, I really shouldn't have, at least I didn't choose the lavender that matches this. I had that option for a pen. I got the pink instead. Okay. CAU. First base, second base, third base. Notice first base. First base, second base, third base. So what I'm going to do is the first base is a C. And so I'm going to look for C on this column right here. And C is right here. So that puts me in this row. This block of rows actually is where our amino acid is going to be. It's going to be somewhere here. Our second base is an A. If you can make that out. So up here we have second bases, and reading through the notes, we've got it. Reading through the letters, you got a U, a C, an A, and a G. So we're gonna go for A. So it's this column. So now we've reduced our area for which our amino acid is gonna be found into this big square right here. Okay. And the third base is U. And so over here, oops, didn't mean to do that. Over here, each block has its own little, the four letters repeated, so we don't have to, we can just look right there. So U is going to tell us the exact line in this block where our amino acid is located. There it is, CAU. And so these are abbreviated. This stands for histidine. So what I'm going to do is I'm going to go and reduce that. I'm going to go to the next slide. Well, actually, yeah, I think I'm going to go to the next slide. So I could write it right there, but I'd be writing through stuff and yuck. Okay, there we go. Come over here and I'm going to write histidine. And I'll try to write out the full word. I'd like to abbreviate because it's faster, but um, a table I give in class for this. And then I always put a little dash that represents the peptide bond. The table I get out in class for this, I think does have it written out. So again, the one I will put in your notes on D2L or supply be for the test if there's not one in your book, should have it written out. Um, if not, it's the first three letters. Basically, I can there's maybe an exception to that, but it's typically the first three letters of them. And so I will, multiple choice, you will have the answers to choose from to get the right answer, to get the right sequence. And so those first three letters should match if I've written out the words and your table just has the abbreviations. Okay, but I'll try to get one that has it for you in the same form. Okay, so our next one, oh wow, okay, GGU. GGU. Okay, first base is G, second base is G, so that's gonna put us down here. GGU right there. And it doesn't matter. On this one, notice that all of the possibilities in this box are gonna be glycine. So this is something probably you should note. Notice that valine has 4 possibilities, proline, a lot of these have 4 possibilities. So there are multiple ways to code for the same amino acid, but you do not find one code coding for more than one amino acid. Okay, so that's kind of important. There's no ambiguity as to which amino acid it's coding for. There are multiple codes for the same amino acid, though. Okay. And then at this point, again, I think probably a good idea would be for you to pause and do the next three without watching me, and then come back and uh, check how you're doing. Okay, if you've got it, if you've got the use of this table down, then you're probably pretty good. Okay, so we have histidine, glycine, adenine, alanine, and a glycine. Again, I just abbreviate the second time around. I can't get that S to look anything other than a G. Okay. Sorry about that. So anyway, that is your amino acid sequence. And again, you can practice this by just any combination, anything you make up. Word of caution on that. Um, Note we've got stops up here. And since you can go back and pause and look at all that, I'm just going to go ahead and erase all the drawings. Okay. Uh, so these, if you run across these codons, they code for stop, and that would tell the protein to stop or the messenger RNA to stop being made. Uh, so that would be the end of the protein. But in a test situation, okay, and I try to make sure this doesn't happen, but I do make errors. Uh, in a testing situation, if I'm asking you to make an mRNA off of a, well, actually, I wouldn't be. If I'm asking you to make an amino acid sequence off of an mRNA, and you run into a stop codon in the middle of the mRNA strand, don't stop. Just tell me it's stop and then keep going, because what it is I gave you a section of messenger RNA and I want you to translate what it codes for. And if there's a stop in the middle, then well, that was supposed to be a stop. But you keep telling me what those codons mean. Okay, um, it's not supposed to happen. Okay, and I think I have gotten that taken care of, and I usually try to double check on tests, but sometimes I'm in a hurry and it's like, yeah, that one's good because I've used that for a few semesters, and sometimes it's not. Sometimes I've gone and grabbed it back off of an older semester where I made a mistake that I didn't catch, and I then perpetuate it. So just word of warning that don't stop at stop. Okay, that's not part of the test. I'm not asking you if you can read it as a protein enzyme making protein would. Okay, just do it. The other odd thing about this is that AUG codes for methionine, or if it's at the beginning of the sequence, it means start. So if AUG is your first RNA codon, then it's going to mean start. Okay. Um, and if it's in the middle of the sequence, then obviously it doesn't, or even at the end of the sequence that I give you, it doesn't mean start there, it means methionine. So that's where you have to look at location, 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 location on AUG. Okay. So those are only things to kind of be wary of about the sequencing on the test. And we're with you on the multiple choice versions. I usually give very short segments. I don't give really long ones. Whereas with the short answer part of the test, I often will make it a pretty long one and things like that. This is more likely to occur on something that is a short answer and you're transcribing it. Whereas with multiple choice, typically I typically don't have the sequence start with this or end with a stop or anything like that. It's just going to be 4 or 5, 6, no more than 6 amino acids sequenced together, and you've got to determine which ones they are. Okay. All right, that's it for this. The main thing I wanted you to understand on this was how a DNA strand is copied for replication, and then how the information from a DNA template gets made into a protein. Okay, the steps involved in the um, the in reading that. Okay, conveying the information and reading it. Okay, so I'll end that here, and I think that'll end off the DNA replication section. Actually, I'm going to take that back. Sorry, got your hopes up. If you shut off already, sorry. Still good. Okay, take that back because I do think I've got one last thing I want to talk about. Since mutations are our main topic here, uh, yeah, uh, or our second main topic. Mutations are where we have a change in the sequence, obviously, and that can be caused by errors during replication, as mentioned at the previous presentation on this. It can involve large regions, or it can be just one base pair gets substituted. And here's an example: the sickle cell anemia protein, for the hemoglobin. Okay, in a normal hemoglobin, this is the sequence. and this is your mRNA, and it codes for, uh, oh gosh, what is it? Glucanine? I think so. I think it's glucanine. Okay. And then with mutant hemoglobin, the sickle cell hemoglobin, yeah, it is a mutation. Well, it's a variation. I mean, almost mutant has such a negative connotation that I kind of hate to call it that because sickle cell anemia has a survival advantage for areas of, or at least historically, has had a survival advantage for areas with high malaria. Um, and that it gives resistance to malaria. It brings with it other health issues and problems that occur, especially as a person gets older, with increased chance of heart attack and stroke in 40 to 45 age range, which today is considered young, but historically, you were a grandparent at that point, and your kids were adult, and you had biologically, for survival of species, served your function. You'd reproduced, or you hadn't, and you weren't going to. you know, uh, and so you reproduced and raised your young old enough to reproduce on their own. So that for survival of species, that's all that's needed. So we want to live longer and want our families to live longer. So it can have some negative connotations and health aspects that it brings on, but it has an advantage too. So it's like So the variable, the variable hemoglobin has this one nucleotide here different. It has an adenine instead of a thymine. So uh, we're going to get a uracil here. And that's going to code, this GUA codes for valine. So one amino acid. Now, sometimes a one amino acid change in a protein will make that protein non-functional. But here it doesn't. It doesn't make it non-functional. It just makes it function differently. It causes the hemoglobin, the red blood cell, to be shaped differently, sickle shaped, instead of the kind of round donut shape. And so uh, it carries oxygen differently, and it flows in the blood differently. And so um, issues with that. Okay. Um, so what we can have, what that is an example of, is that is a base substitution. Okay, the number of bases stayed the same. It's just one got swapped out for another and it's substituted. But we can also have a mutation where we have a deletion. Okay, a base just got swapped out, and then everything frame shifts, and those are called frame shifts. It's called a frame shift mutation. Everything frame shifts over one, and so from there on out, you have every single amino acid is going to be different typically, uh, unless by random chance. Yeah, but usually if the first letter changes, you're definitely going to get a different amino acid. Okay, so even in those where all four were the same, the first letter is the same. The second and third letters, usually the second letter is the same. First letter, second letter is the same. Okay, and then the third letter can vary. So if you frame shift completely over, it's usually going to give you a different amino acid from there on out. So that's a much more drastic type of mutation than a single base change, which depending on where it occurs, may put a different amino acid in there. Now, in a lot of cases, if your base mutation occurs at the third slot, okay, the third slot here of the triplet, no, no amino acid change will occur. Okay, so that's a no effect mutation. Kind of interesting. Okay. Uh, Along with deletion, we can get an addition. We can get a base addition, which is going to slide an extra one in. And again, that'll frame shift everything the other direction and results basically have the same result as the deletion, just everything got shifted the other way, but you wind up with all the amino acids after that being different. Okay, so these are typically much more devastating as far as whether the protein will function or not, and usually means that the cell this occurs in will no longer function, and if that's in uh, the genome of a single-celled organism, or an organism, the single cell that's going to mitotically divide and form a multicellular organism, then it won't survive. Generally speaking, obviously there's cases where or we expect there's cases where this happens and something totally new and different happens and it's great and wonderful, but that would be very rare. Okay. And that will end it.